Hello, dear zoos, learners, multicellular organisms, Homo sapiens sapiens. Greetings of the day. Welcome back to our zoology class. What are you searching for? Are you searching me? Yeah, I will be in front of the camera now. Yes. How are you all? Okay. In this video module, we are going to discuss the zoology topic. Interested one, epitopes of antigens. Epi upon topes, special type of structures or bodies. Let's see what they will do. What are they doing upon something? Upon in the sense where they are present, why they are present, what purpose they are present. Let we discuss. Before going to epitopes, we will come to know that antigens, antibodies, anti in the sense negative, gens in the sense generating or life, etc. Then bodies in the sense shapes or structures, antibodies. So anti, against. So these bodies are against of something. So for what these against in the sense antigens. So antibodies are against the antigens. Are you clear? So RBC, we knew RBC in the sense red blood cells or erythrocytes. Production of RBC is known as erythropoiesis. Production of WBC is known as leukopoiesis. Production of thrombocytes or platelets is known as thrombopoiesis. So RBC is a type of formed element of blood. Blood is a fluid connective tissue. We knew that. RBC, this is a biconcave. Matured RBC does not contains nucleus. Immature RBC contains nucleus. Okay. So on this RBC, generally one pigment is present, hemoglobin, respiratory pigment. Due to that pigment, our humans and animals body blood contains red color. Okay, this one we knew already. On this RBC, apart from hemoglobin, one special protein is present. That special protein is known as antigen. Here, antigens two types, antigen A, antigen B, based upon their chemical bonding and structure. Antigen A, antigen B. Okay. Then coming to the plasma. What is plasma? Straw colored fluid which is present in the blood, which doesn't allow the blood corpuscles combine together, bind together, collapse together, clumping together. So that fluid is known as plasma. In this plasma, two more special type of proteins are present. Those proteins are known as antibodies. So here antibodies also categorized into two categories, antibody A, antibody B. So dear everybody, juice learners, now this is clear, I think so for you. Antigens are separate, A and B. Antibodies are separate, A and B. Antigens present on the RBC, antibodies present in the plasma. Are you clear? Then serum. What is a serum? We knew after collecting the blood in a test tube, within a few minutes, we could observe a transparent fluid in that test tube. That is known as plasma. Generally, after collecting the blood in the test tube, we could observe the fluid, that is plasma, slightly transparent, actually straw color. Then after clotting of the blood, the transparent fluid present on the clotted blood surface is called serum. Best example, after killing or cutting the head of these goats and sheep during the festivals or the devotional purposes in the villages, what it is called in Telugu, Bali, Peddamma Talli or something, Potulaya Swami, whatever it may be. So after that Bali process, that blood is flown on the surface, or else the blood is collected in a bowl in the butcher shop or that mutton shop, then we could see in transparent fluid on the blood surface, that is known as serum. So serum is very 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 important. Why? Because in that serum these antigens and antibodies also present, especially antibodies are present. So during the blood transfusion from one person body to another person body, serum plays major role. Remember everybody. Okay, then we knew now antibodies types and antigens types and where they are present. Okay, then coming to epitopes. Epitopes in the sense, on the antigen surface special receptors are present. So those receptors receive the antibodies for the binding purpose or attachment. So these receptors, wherever they are present on the antigen surface, that exact location is known as epitopes. Are you clear? So you knew already cell, cytoplasm, nucleus, inside the nucleus, nucleoplasm. In the nucleoplasm, thread-like structure is called chromatin fiber. During the cell division, this chromatin fiber condensed, then they will be converted into chromosomes. On the surface of the chromosomes, genes are located. Then where the genes are located? On their exact location called gene locus. Are you clear? So same like gene locus, here on the surface of the antigen, that is called epitopes. Simply, where our human heart is located, in the chest cavity, exactly in the mediastinum. So here we can consider that mediastinum as epitope. Then one more example, our human brain is located inside the skull, especially that cranial cavity. We can consider that cranial cavity as epitope. Are you clear? Very simple. You all are the students. You have a classroom. In that classroom you have desks. In each and every desk, particular student will be sitting. Yes or no? Every day that particular student is sitting. So if we consider that desk is antigen, that student is antibody, where he is sitting, he or she is sitting in her or his particular place every day. So that particular place in that desk is known as epitope. Are you clear? Then do you have an idea of that one famous dialogue? Which dialogue? Black sheep, Vasi, meow. Yeah, from which movie? Yeah, Robo. So superstar movie, from that Robo movie also, when the hero Rajinikanth entered into the group of the artificial robots, so that person name is Vasi actually. So this villain robo will try to find out that humanoid robo. So finally, after several testings, it will pass the order to the all robots to rotate neck. So all robots will be rotated the neck. Only that particular humanoid robo, that hero, unable to rotate the neck. Then finally, this villain robo will find out that humanoid robo. So here, these all robots together known as, just example, antigen. That antigen has one particular site. Site in the sense, one robo, it is unable to rotate its neck. That is called humanoid. So here, this villain robo is, villain Rajinikanth is the antibody. So this antibody found that hero robo. So this antibody found that epitope. Are you clear? So antibody required one specific site to bind with antigen, that is known as epitope. Don't be confused. Like this, several examples we can be taken. Take an example, you are going for a trip. You have booked a ticket. So here that bus is antigen, you are the antibody, then you have booked a seat number, that ticket number, you should be allowed to sit in that particular seat, yes or no? Other seats already booked by someone. In the sense, someone or other antibodies, you are specific antibody, you have the special seat number to sit, so that seat number is nothing but epitope. So you are allowed to sit in that only particular seat number, that is known as epitope. I think so everybody now you are clear. So such type of many examples can be taken, but we don't have time, sorry, we will continue. Then so clearly we knew now antigen. On the antigen surface special receptors are present. Those receptors are the special sites which they will receive the antibodies for the further action, that is called epitope or epitopes. Generally single antibody can be bind with a particular epitope. So sometimes many antibodies also they have capability to bind with single epitopes. Are you clear? Single antibody can bind with single epitope. Sometimes different antibodies can bind with single epitope. Not only single, many antibodies also can combine with single epitope. So on the antigen surface several epitopes will be located or fixed or arranged for the binding of antibodies. So here antibodies are different types, 5 main types: IgA, IgD, IgM, IgE, IgG. Ig in the sense immunoglobulin. So antibodies are generally called immunoglobulin. So here to remember these 5 types of antibodies, one small and simple trick is damage. DAMAG, damage. IgD, IgA, IgM, IgE, IgG. So damage 5 antibodies. Ig means immunoglobulin. Remember. So dear Zeus learners, sometimes very rare on the antigen, take an example, 5 antibodies are binded with 5 isotopes. Listen clearly. On one antigen, 5 antibodies are binded with 5 isotopes. If such type of antigens are more in number, are you clear? Already with one antigen, 5 antibodies are binded. Same like that antigen, few more are there. Best example. So these few more or else remaining antigens also they are having the same isotope. So here what will happen? These antibodies automatically attack the remaining antigens. Are you clear? Generally, single antigen has 5 antibodies binding site. So those 5 antibodies binding sites also same for remaining few more antigens. So here what will happen? Same antibodies whichever binded with one antigen, those antibodies automatically attack or kill or function or control or dominant remaining antigens also. Why? Because remaining antigens also they are having the same binding site, right? So this phenomena is known as cross-reacting antibody mechanism. Are you clear, dear Zeus learners? So, cross-reacting antibodies mechanism in the sense attacking different types of same antigens by the few antibodies. Are you clear? So, here B cells, T cells, natural killer cells. So, these are the different types of cells which are present in the immune system of our human body or else animals. B cells in the sense B lymphocytes, T cells in the sense T lymphocytes, NK cells in the sense natural killer cells. So, among these all, first place B lymphocytes. Why? Because these B lymphocytes are responsible for activating or production or guidance of the antibodies. Take an example. One person body is attacked due to the coronavirus. So, after examine by the experts, the vaccine will be given. So, that vaccine is, what is meant by vaccine? Dead and weakened microbes are called vaccine. Again, based upon the chemical content, vaccines are again 6 types. We will discuss in one more video, types of vaccines. So, here that dead and weakened microbes are injected into the body, that is called vaccination, that is also called immunization. First time, whenever the coronavirus is entered into the body, these B cells, they were recognized, scanned or taken the Xerox copy, whatever it may be, earlier. So, second time whenever the real coronavirus will be entered into the body, then automatically these B cells activate the antibodies regarding the particular coronavirus in the body, whichever already generated, then the body is protected. That immune system fight against that coronavirus. Not only coronavirus, any type of foreign body, nothing but antigens. Whichever the foreign body which activates the antibodies known as antigen. Are you clear, dear students? So, according to our discussion, this is a simple topic, but if we continue, many links we will be getting. So, video will be prolonged later. In our next video, we will meet with one more beautiful topic from our zoology subject. Till then, Homo sapiens sapiens. Take care. Good luck.